Inflammatory bowel disease, which is a chronic autoimmune disorder that affects the digestive system, as well as the classification given to both Crohn's disease and ulcerative colitis, is a disease characterized by inflammation of the gastrointestinal tract. IBD affects over 200,000 Canadians, the prevalence of which has increased nearly 50% as of the year 2023. Treatment options include anti-inflammatory and immunosuppressant medication, and ways to test for the disease involve a series of laboratory tests with biopsies, imaging, and oftentimes endoscopies. Laboratory models of this disease have been made to study more complex processes through studying inflammatory markers from biopsies and serum tests. These tests are typically available in more advanced settings, in part due to their advanced nature, and as such, the research capabilities of more intermediate work, especially in that of higher education institutes, is very limited. To try and tackle this problem, I set out to develop a model of the intestinal tract simulating IBD in the same vein as simplified chemical blood serum antibody tests. I accomplished this using intestinal or colorectal cells and immune cells. The idea behind the model was to find out if, first, the intestinal cells would remain viable in the presence of the immune cells in a co-culture model, and second, produce the inflammatory markers associated with the diseased GI tract. Developing the co-culture model began with the culture of colorectal CACO2 cells and immune THP1 cells in separate plates, the former of which was grown in transwell inserts. After 2 weeks of growth and differentiations into intestinal monolayers and macrophages respectively, the intestinal cells were then moved into the THP1 cell wells to begin the bacterial lipopolysaccharide treatment of the immune cells for 24 and 48 hours of stimulation. The viability of the intestinal cells in the model was measured using an NTT assay, where I observed no significance of cell viability. With that knowledge, I prepared a second plate, wherein I grew the colorectal and immune cells in separate well plates and treated the CACO2 cells with the cytokine-rich supernatant of the immune cells. Despite this new model displaying a significance in viability, I had decided to move on to the quantitative PCR test with the co-culture model, as it was a direct simulation of the GI tract. I prepared one more plate following the same procedure as the co-culture well insert model to perform qPCR, and interestingly, found significance in the expression of tumor necrosis factor alpha, or TNF alpha, which is the pro-inflammatory cytokine associated with IBD, in the comparison between the 24 and 48-hour mark. Knowing now that the cells within the model remain viable, as well as observing the significance in the expression of the associated pro-inflammatory cytokine in the extended treatment groups, I concluded that the model held a viable potential for future and extended use in IBD simulated studies. Further optimization of the model, as well as utilization of a more sensitive testing method, such as an ELISA test, to better measure the expression of the cytokine in the intestinal cells, may better enforce or contradict the true viability of the model and its potential for future use. To conclude, the viability of the cells in the co-culture model, alongside the expression of the IBD-related inflammatory cytokine, TNF alpha, has proven its potential as a viable and more accessible model for higher education institutions, as well as lab research, which could pave the way to better understanding the true cause of this disease and working towards finding more effective treatments and possibly cures.